Hi and welcome everyone. FluGuide recently released phase 2b topline data from its trial with FG001 in high-grade glioma. And today to discuss the results, we have the company's CSO and CMO Andreas Kjær and the company's CEO Morten Albrechtsen. Welcome.
Thank you, Christian.
Thank you for having us. So I would start out and just present a little bit. We are very excited about sharing the data of the top-line results of the recently concluded study in aggressive brain cancer. So just to go to remind everybody about what we are really doing in FluGuide, we are powering precision surgery, so we are doing optical guided surgery. And the way it works is that we have a fluorescent probe. It's injected into a blood vessel, then it distributes in the body, binds to the cancer, and is removed from the rest of the body. In this way, cancer will light up and surrounding tissue not. And that can be used for guiding the surgeon. And that is what we have illustrated here, both for telling the surgeon what to remove, but also for the surgeon to check if anything was left behind. And if surgery is done better, the outcome for patient is better. So this is what our technology builds on. And in this study, we looked at aggressive brain cancer, and the primary objective was to see the efficacy of FG001. So in patients with malignant glioma undergoing surgery. And we then compared it, and I come back to the design, with another arm where we gave 5-ALA that in some places of the world is currently standard of care, but not in all jurisdictions. And the primary endpoint was to evaluate how many patients where fluorescent-guided surgery or FG001, the fluorescence, could identify extra cancer tissue. So in how many patients did the surgeon think they removed everything? Then when they turned out the special light you need to see FG001, then it turned out that it was actually sitting tissue there that lighted up, and that turned out to be cancer tissue when it was removed. So better, how many patients did you make a more complete resection, more complete surgery when you were guided by the optical signal? And always when you do studies where you compare, where you have two different treatments or two different drugs, in this case, FG001 and 5-ALA, then you want the groups to be the same. If the tumors were different or they had different age, then it wouldn't really be comparable. And I won't go into any details here, but this is the normal check that the two groups actually are very much alike. And it actually also went for their tumors that were very much alike in size, but we haven't presented it here. You can also see that each group had 12 patients. So the trial design, just to take you through that. So we had 24 patients in total, and they were randomized. So like a lottery, the surgeon couldn't say, I want this patient in this group, I want this patient in another group, but it was like a lottery. Randomly, they were divided to either getting FG001, our compound, or 5-ALA. And then surgery was performed in white light. So without really being able to see the optical guidance yet, then the surgeon said, I'm done. And then they could turn out on the special light needed to see these optical guidance. And then the readout was in how many cases did the surgeon see? Well, there was actually something left behind that lighted up, removed it, and it was histopathologically, so by the pathologist, confirmed that it was actually cancer tissue. And the results were amazingly good, and actually for both compounds. So in all 24 patients, and in particular for FG001, because this has not been studied in this way before for FG001, in every patient, the optical signal found extra tissue. So you could also say that if you are not using optical guidance, in all patients you would overlook extra tissue that would have been able to be removed if you use the optical guidance. So our primary endpoint could not have been met stronger in this case. And of course, we are quite excited about this going forward. So this was a prerequisite that it worked well. Another part of the study that was also studied in the earlier brain cancer study is, of course, always to look at the safety. So you build your data on safety. So every time you do a new study, you add on the number of patients where you can see that it's safe. And the more patients you have seen that in, the more sure you can be that it's safe. And what we can see up here is AE is what is called adverse events. And you do surgery in the brain, so a lot happens just because you do the surgery. It is quite a big manipulation you do with the brain. But what is interesting is to look at what is called related adverse events. So adverse events that are deemed related to the drug, to the compound. And here you can actually see that in total, so that's for the 24 patients, 12 in the FG001 group and 12 in the 5-ALA group, there were a total of 13 related adverse events. But in fact, you can see only 2 of these 13 were seen in the FG001 group. So even though this was not powered to do a statistical comparison, we can just see in these preliminary data at a very low number of related adverse events was seen in the FG001 group. So we now know that it was safe, as we have seen before. We know the surgeons in all cases found extra cancer tissue. Then another readout we always do is to describe how strong is the contrast. At the end of the day, it's the contrast the surgeon acts upon. So the contrast is the difference in light between the tumor and the surrounding. And we call that the tumor to background ratio. If that was 1, it would be the same light in tumor and surrounding, and you couldn't use the method for anything. Normally, as a rule of thumb, you want something that is 1.5 or higher. And what we found is that on average, we were actually above 5. That's the line in the middle, but you can see all the dots are the single patient in FG001, and you can see in every case it's above 2. So it's better than the rule of thumb also for the single patient. So very encouraging, and what is a relevant signal so the surgeons can act upon it. So taking it together, the top-line results from our study showed that all patients receiving FG001, 12 out of 12, had additional cancer found by optical guidance, underscoring the real need for optical guidance. 5-ALA also demonstrated that extra tissue could be found in all patients. We found that FG001 was safe and well tolerated, with only very mild related adverse events like nausea and in that line, and that FG001 lit all the cancers up with the tumor to background ratio larger than 2, clearly above the rule of thumb, what you need. Then we looked at many other measures, sensitivity, in how many cases do you identify the tumor tissue? If it's not lightening up, then it should also not be tumor. And all these measures were studied for both of the compounds, but because there are only 12 in every group, we found that there was no difference between any of these measures. But again, you need a larger study if you want to see or detect subtle differences. And finally, the pharmacokinetics showed that the plasma half-life, the half-life in blood is 13 hours, and that we have a very high maximum concentration of the compound. So everything very encouraging to move forward. And then I hand over to Morten.
Yes, thank you very much, Andreas, for presenting this data. I just want to put an reflection on it on commercially. So this data is really, as Andreas has been through, a landmark result for FluGuide. So we have a clear path to the market for one indication in one jurisdiction, at least. And we can, as you can see on the graph, you can see the number of patients in the US. There's about 15,000 new patients with high-grade glioma every year. And there's a lot of, unfortunately, other brain cancer that is coming in, being diagnosed every year. You can see low-grade glioma, meningioma, other brain cancer and metastases. So that's one way we can grow. We can grow into other indications. You know, we recently have just announced a very good result in head and neck, and also before summer in lung for thermal therapy, and we go into other geographies. So for us, we're very pleased with this result. We have one indication, one product, at least one jurisdiction, and from here is just optimization. So thank you for that.
Thank you so much for that great presentation. Now let's get to the Q&A. To start with, as you mentioned, both FG001 and 5-ALA in all patients enabled the surgeon to remove additional cancer tissue. Does that mean that all cancer tissue was removed in the end, or was there still some residual cancer tissue left?
I can start with this. The design was made to see whether extra correct tissue was moved. We did not investigate whether all cancer was removed. In brain cancer, it's very difficult. The way the poor prognosis this particular cancer has makes it likely that there are islets of cells that are left behind with and without. But still, the better you do the resection, the better outcome it translates to. When we look into other cancers, even though it's brain cancer we present today, like head and neck cancer, then it's definitely a question of really removing everything. So it's very encouraging. In this particular case with brain cancer, it is often very difficult. And there were also many of these patients where you could not remove everything, even though you could see it lighted up, because the brain is precious functions. So if you know that it's very close to something that would lead to a palsy, or something that would lead to that you would lose your ability to speak, then the neurosurgeons need to respect that. So in I think it was around half the cases, it was not really possible to go in and do the full resection. So it's a little bit of a special case. So the important thing is you remove more correct brain cancer. When you have a delineation, you also do not remove, you do not work so much in the blind. So one of the positive things can also be that you actually remove less, because you can see it's not lightening up here. Otherwise, you would have to make a margin to be sure. It might be difficult to show in a clinical trial, but we believe that you will do a more gentle surgery. Remove more of the correct cancer tissue and less of the healthy tissue, and thereby being able to preserve function better.
Very interesting. Now, you've already touched on it a little bit in your comment right now, but looking at a potential phase 3 trial, how do you think FG001 could deliver better results compared to 5-ALA, as this trial wasn't really designed to do so? And is that even relevant, i.e. are you planning to, for example, go for a superiority approval, or could it also be a non-inferiority approval?
Yeah, I can start with that. It's a thing we are designing right now. In some geographic areas, 5-ALA is not standard of care. So it can both be a comparison with 5-ALA, in some cases maybe not. But what is important of this readout, maybe many would say, yeah, but now you have something that works really well, but it seems that it's very much like 5-ALA. And as we see it, it's great if both technologies are efficient in identifying extra brain tissue. But if they are on, even if they are on par with that, then FG001 compared to 5-ALA carries a lot of other advantages. For one thing is that we can see in depth and it simply comes with the physics. We use near-infrared light, so we can see centimeter down in the tissue, whereas 5-ALA can absolutely only see a very, very thin layer. So for the surgeon, they lose this three-dimensional impression of where the tumor is. This is one thing. The other thing is that 5-ALA has to be given 4 to 6 hours prior to surgery, whereas our compound is typically given longer the day before. We recently went to a conference in the US and discussed with a lot of key opinion leaders, and it actually turned out that practice somewhere in some places in the US is that the first patient in the morning is actually not operated with 5-ALA because they don't want to administer the compound in the middle of the night. FD001 is easily administered basically every time a day, one day before, so it's a much more convenient workflow. So in this case, the surgeons would say, with this workflow, we would not have to let some patients not being operated in 5-ALA. So we believe that that there are a lot of advantages in it. It could also turn out, this is not a thing we have support for in current data, but it could well be that the depth you see, the way it works, it would also be a better learning curve for young surgeons. You have to remember this is very experienced surgeons that use these technologies. And we believe the way our compound lights up might be easier to learn. But of course, that remains to be proven. So in many ways, we would be good even if they worked on par.
Got it. And in a potential phase 3 trial, if you have already determined that, what would be the primary endpoint? Would it be similar in terms of, for example, change in surgical strategy? Or would you also measure, for example, patient survival?
I can take that. We are discussing with regulators as we speak with the right endpoint. There's 3 different endpoints that is used in trial. One is the exact same as we did here. That's the most recent approved products. There's one looking at the 5-ALA was approved by accuracy, so the sensitivity, specificity, positive and negative predictive value, and as well some survival data they submit, but actually was approved by accuracy. So the endpoint will be somewhere in between, and we are currently discussing this with the regulators, what would be the optimal endpoint. So we haven't decided that yet, but it will most likely be additional tissue removed at the end of surgery.
Understood. Now, we've also received a number of questions before this session, and they mainly evolved around, the potential of photothermal therapy, and specifically, as you said, in several patients, additional cancer tissue could be removed, but not all cancer tissue was resectable because it was too close to healthy brain tissue. So we've received a lot of questions about why not combine surgical guidance with photothermal therapy now to be more able to clearly prove an advantage over 5-ALA. What can you say around that?
I can maybe start, Morten, with just saying it was actually one of the advantages I forgot to mention, because you cannot heat, there's no photothermal effect of 5-ALA. So as we said, if it delineates the tumor on par, we also have this layer that the fluorophore we use, ICG, is very good in converting laser into heat. So we can burn away the tumor cells, which cannot be done with 5-ALA. So it's a thing we pursue. Research-wise, we are deep into this field. And what, of course, remains to be decided as a strategy at the company level is, of course, is it something that is combined? Is it something that is a follow-up study? But the concept, of course, is that once you have administered the FD001, you can do surgery after it. And those parts, and even the parts you do not see, you will be able to heat on specifically. So you would heat the cancer cells and not the surrounding tissue. And how that should be positioned exactly and when we do it, I think it's fair to say that's internal discussions. No doubt that we will leverage on that capability of our compound, which has been shown in animal experiments already in mouse models that with human cancers, that it works very well to burn away the cancer tissue.
Got it. And can you say anything more about, potential development timelines if you would go for surgical guidance versus photothermal therapy? Could you go to a phase 3 trial in both, or would additional trials be required?
Yeah, I think if I may take that one. I think one of the important things for us has been, since we start the development, is that we would like to be predictable. Then rather take it a little bit more slowly, but be predictable. And one of the things for photothermal therapy is that if we jumped into a too advanced study too quickly, if it went well, I mean, it would be fantastic. But there's a high risk that we will fail, not because it didn't work, but just because we did the study wrongly. So for us, it's very important to do that in stepwise approach to it, and that's also speak for, as Andreas mentioned, why we would do most likely a pre-study before jumping into later stage development with this. I mean, that's important to de-risk it. You still have a quite high risk on us, so if we de-risk it a little, we actually increase the value enormously. So that is a key thing for photothermal therapy, that's de-risking.
Understood. And now 2 remaining questions that I would like to discuss. First of all, we've already touched on it a little bit, and you may not have the answer just yet. But when it comes to a potential phase 3 trial, irrespective of what modalities you use, from a current strategic perspective, would you think that market uptake and commercial potential versus the risk would be optimized if you go for a non-inferiority approval or a potential superiority approval?
Well, I think that commercially, the benefit we will get for superiority approval in terms of price and reimbursement will not be worth the effort. Then it will be much more worth the effort to put that additional cost into photothermal therapy approach and really take it to the next level. So the key reason for us selecting high-grade glioma is that it's with a high likelihood with offer an indication where we can get predictable into the market quickly and then grow from there. That's the kind of the idea with high-grade glioma. Photothermal therapy is a significant step forward in terms of a much higher potential price we can get for it, because we also add more value to the patient, proven by the studies we will do. We will take it that way.
Got it. And then when it comes to primary endpoints, to get back to that discussion really quickly, as we discussed, it could be either, for example, removal of additional tissue, or you could also have some survival measures. In your previous trials, patient inclusion was always really short, about 48 hours, if I remember correctly. Depending on which outcome measure you choose for a phase 3 trial, how would patient inclusion develop? I guess for just removing additional tissue would be similar, but how would it change if you would have to follow patient survival?
Yeah, if I can take that. Unfortunately, and that is the sad fact about high-grade glioma or glioblastoma, as you might recall, the median survival, the average survival is only little more than 1 year. So for this reason, in a clinical trial, you do not need to follow for a very long time to see a potential difference. Whereas you had some very benign cancer, you would have to follow for a long time. So typically 6 or 12 months would be the maximum you would have to follow in this particular indication. And typically you would look at what is called recurrence-free survival. So basically you would look at when does the tumor regrow.
Understood. Perfect. I think that wraps up all the questions that we had. So Andreas and Morten, thank you so much.
Thank you for having us.